Hi, I'm Arely Perez from the SIP North Mexico, and today I'd like to share work on the functional compartmentalization of bacterial communities associated with the native coastal desert legume Sortamus mori. We already know that plant-associated microbiomes play an important role in nutrient acquisition and stress tolerance, especially in extreme environments such as coastal deserts. So a question that motivated this work was whether these bacterial communities rely on functional redundancy, where many different taxa perform the same ecological functions, or whether they show functional partitioning, with different functions being concentrated in different compartments of the plant. To answer this, we studied the culturable microbiome associated with Sortamus mori, a native legume of harsh environments such as the coastal dunes of El Mogote, Baja California Sur. We isolated 90 bacterial strains from two fractions, roots and soil, and then from these we selected 45 representative isolates, and we screened each isolate for 7 plant beneficial traits, including salt tolerance, to which we designed an assay to identify allotolerant bacteria in a gradient of LV agar with salt. And then we also analyzed the phosphate solubilization, nitrogen fixation, SIDE4 production, and antagonism against bacterial and fungal pathogens. We also identified the isolates using 16S ribosomal RNA sequencing and compare both their taxonomic relationships and their functional profiles. Here, our phylogenetic trees showed that beneficial traits are not restricted to specific bacterial lineages. Instead, these functions are distributed across multiple taxa, suggesting functional redundancy. But when we compared the root and soil communities, we found that although both compartments, root and soil, showed similar levels of functional diversity, they differ in functional composition. Root communities were enriched in functional specialists, while soil communities contained more multifunctional bacteria. Among all the traits we evaluated, phosphate solubilization was the most common, and antagonism against Clybucter michiganensis was the only trait significantly associated with habitat. Overall, our results suggest that in the coastal desert legume, microbial compartmentalization is driven more by the combination of functional traits than by taxonomic identity. This highlights the importance of evaluating microbial functions when selecting candidates for biotech applications.